Let us see about microbial fermentation. Fermentation does nothing but microorganisms, like all living things, metabolize nutrients in order to grow. By metabolizing nutrients, microbial cells produce ATP and other substances. If metabolism is carried out under conditions where oxygen is absent, it is called fermentation. Fermentation is historically important method of food preservation. Acids produced in yogurt, cheese and pickled vegetables inhibit growth of many spoilage organisms and foodborne pathogens. What are fermented foods? The foods that have been subjected to the action of microorganisms or enzymes in order to bring about a desirable change are called fermented foods. Numerous food products owe their production and characteristics to the fermentative activities of microorganisms. Fermented foods originated many thousands of years ago when presumably microorganisms contaminated local food. Let us see about significance of fermented foods. Microorganisms cause changes in the foods which helps to preserve the food, extend shelf life considerably over that of the raw materials from which they are made, improve aroma and flavor characteristics. It also increases its vitamin content or its digestibility compared to the raw materials. Various types of fermented foods are bread, fermented meats, sourdough bread, fish sauce, pickled vegetables, mushrooms, soya sauce, wine, fermented milk, and cheese. What are the microorganisms that are involved in preparation of fermented foods are lactic acid bacteria and yeast. Group of organisms most frequently used for production of fermented foods are the lactic acid bacteria. They require amino acids, B vitamins, nitrogenous base and an optimal pH for growth. Yeast are also used in the food industry for fermentation. Let us say about lactic acid bacteria. This is a group of fermentative organisms. This group is comprised of 10 genera of gram-positive bacteria. It includes Corynebacterium, Enococcus, Enterococcus, Pediococcus, Lactococcus, Streptococcus, Lactobacillus, Vagococcus, Lactospira, and Leuconostoc. Related to this group are genera such as Aerococcus, Microbacterium, and Propionibacterium. Let us see about lactic acid bacteria. This lactic acid bacteria can be divided into two groups based on the end products of glucose metabolism. Those that produce lactic acid as a major sole product of glucose fermentation are designated as homofermentative. Those that produce equal amount of lactic acid, ethanol and CO2 are termed as heterofermentative. The homolactics are able to extract about twice as much as energy from a given quantity of glucose as the heterolactic. Lactic acid bacteria growth. The lactic acid bacteria are mesophils. They generally grow over a temperature range of about 10 to 40 degrees centigrade, up to optimum between 25 and 35 degrees centigrade. Some can grow below 5 and as high as 45 degrees centigrade. Most can grow in the pH range from 4 to 8, though some as low as 3.2 and as high as 9.6. Starter cultures. Traditionally, the fermentative microorganism came from the natural microflora or a portion of the previous fermentation. In many cases, the natural microflora is either insufficient, uncontrollable, and unpredictable, or is destroyed during preparation of the sample period to fermentation. A starter culture can provide particular characteristics in a more controlled and predictable fermentation. Lactic starters always include bacteria that convert sugar to lactic acid, usually Lactococcus lactis, Lactococcus cremoris, or Lactococcus lactis bior acetyl lactis, where flavor and aroma compounds such as diacetyl are desired. The lactic acid starter will include heterofermentative organisms such as Leuconostoc citrovorum or Leuconostoc dextranicum. Functions of starter cultures. The primary functions of lactic starter is the production of lactic acid from sugars. Other functions of starter culture may include the following: flavor, aroma and alcohol production, proteolytic and lipolytic activities, as well as inhibition of undesirable organism. Good starter culture should convert most of the sugars to lactic acid, increase the lactic acid concentration to 0.8 to 1.2 percentage, drop the pH to between 4.3 to 4.5. What are the factors influencing growth of microorganism in foods are intrinsic factor, extrinsic factor, factors that combine to determine which microorganism grow in a particular food and at what rate. Intrinsic factors. Multiplication of food greatly influenced by inherent characteristics of food. It includes water availability, pH, nutrients, biological barriers and Antimicrobial chemical. Water availability. Foods vary dramatically in terms of water availability. Fresh meat and milk have high water contents. It supports microbial growth. Butter, bread, nuts and dried foods have low water availability. Water activity is used to designate amount of water available in foods. Pure water has water activity of 1. Most bacteria require water activity of above 0.90. Most fungi require water activity of above 0.80. pH, it is important in determining which organism can survive and thrive on specific foods. Many microorganisms inhibited by acid conditions. Exceptions include lactic acid bacteria, fungi able to survive at relatively low pH. Most acid foods spoil from fungal contamination as opposed to bacteria. pH can determine bacteria's ability to produce toxin. Toxin production of many organisms is inhibited by acid pH. In nutrient present in food determine organism that can grow in foods. Biological barriers, rinds, shells and other outer coverings help protect food from microbial invasion. Microorganisms will eventually break down coverings and cause spoilage. Antimicrobial chemicals. Some foods contain natural antimicrobial chemicals that inhibit growth of organisms responsible for spoilage. Next one is extrinsic factors. Extrinsic factors include storage temperature as well as atmosphere. Storage temperature affects rate of microbial growth. Below freezing, water availability is significantly decreased. Water crystallizes and is unavailable, halting microbial growth. Low temperature, enzymatic action is very slow, results in inability of microorganisms to grow. Extrinsic factor, other one is atmosphere. Presence or absence of oxygen affects type of microbial population. Obligate aerobes cannot grow under anaerobic conditions. Obligate anaerobes will grow in anaerobic conditions, including certain foodborne pathogens. Let us see about fermented milk products. What are the products that are prepared by fermentation? Production of fermented milk products do not rely on naturally occurring lactic acid bacteria. Starter cultures are added to milk. Strains are carefully selected to produce desirable flavors and textures. Starter cultures must be carefully maintained and protected against contaminations. Cheese productions. Cheese is one of the product made from milk. It can be made from milk of variety of animals. Cow's milk is most common. Cheese is classified as very hard, semi-soft and soft. Classification. It is also based on percentage of water content. Cheese production. Steps involved in cheese production. First, milk is pasteurized and inoculated with starter culture. Culture causes milk proteins to coagulate. Coagulated proteins are called curd. This curd is heated and cut into small pieces to facilitate drainage of liquid waste. That waste is termed as whey. Most other cheeses undergo further microbial processing termed as ripening or curing. That cottage cheese is an unripened one. Enzyme renin is also added to fermenting milk to hasten protein coagulation. Curd salted after whey is separated out, pressed and ripened to encourage changes in texture and flavor. Ripening can take weeks to years. Longer ripening produces more acidic, sharper cheese. Certain organisms produce certain characteristics. Propionibacterium, it is involved in the production of Swiss cheese. Penicillium roqueforti, it is involved in the production of Roquefort cheese. This diagram shows the production of cheese, that is milk is fermented by lactic acid bacteria. Lactic acid is produced, thereby it lowers the pH, which also coagulates milk protein, and by means of other processing steps cheese is produced. Next one is production of yogurt. Pasteurized milk is concentrated slightly, then inoculated with starter culture. Mixture is incubated for several hours at 40 degrees centigrade to 45 degrees centigrade for several hours. Thermophilic bacteria grow rapidly at higher temperature, produce lactic acid and other end products. It also contributes to flavor. Control in incubation ensures proper level of acid and flavor compounds. Next one is acidophilus milk. Sweet acidophilus milks retain flavor of fresh milk because it is not fermented. Culture is added immediately before packaging. Bacteria are added for certain health benefits. Main benefit is it prevents and reduces severity of some diarrheal diseases. Next one is pickled vegetables. Pickling originated as a way to preserve vegetables, particularly cucumbers and cabbage. Pickling causes naturally occurring lactic acid bacteria residing on vegetables, unlike fermentation of milk products which relies on starter culture. This diagram shows the production of sauerkraut. It is cabbage. It is inoculated with lactic acid bacteria. It produces lactic acid which lowers the pH, as well as it also produces carbon dioxide which creates and maintains anaerobic conditions. Thereby fermentation takes place. It results in the production of sauerkraut. The next one is fermented meat product. That is semi-dry sausages. It is one of the fermented meat product. Is prepared by adding 3% salt, sugar, seasonings and nitrate to cubed beef. Pediococcus cerevisiae is then added and fermentation is carried out at 4 degrees centigrade until the pH drops to 5.3. The product is then cooked to an internal temperature of 68 degrees centigrade. This diagram shows the production of sausage. Meat is fermented by means of lactic acid bacteria, that is Pediococcus cerevisiae or Pediococcus acidilacticae and Lactobacillus plantarum or Micrococcus variants. Thereby it results in the production of lactic acid. Similarly, it lowers the pH and which helps in flavor and color development, as well as texture is also developed, and other processing steps are done. Thereby it results in the production of summer sausage. Next one is alcoholic fermentation by yeast. Some yeast ferment sugars to produce ethanol and carbon dioxide. Yeast are also used to make variety of alcoholic beverages as well as vinegar and bread. Alcoholic beverages include wine, beer and distilled spirits. Production of wine. It is a product of alcoholic fermentation of naturally occurring sugars and juices of fruit. Most commonly, grapes are used. Commercially made wine produced by crushing selected grapes. Stems are removed and solids are collected. Entire grape is used in red wine. Juice only used in white wine. Solids removed after 1 day and juice is fermented to produce rose wine. This is the steps that are involved in production of wine. First one is fermentation must be carefully controlled to ensure proper reaction. Sulfur dioxide is added to inhibit growth of natural microbial population. This converts alcohol to acetic acid and most responsible for spoilage. Fermentation process is initiated by addition of selected strains of yeast. At completion of fermentation, wine is then aged in oak barrels. Wine is filtered for clarification and then it is bottled. Next one is production of beer. Production of beer is a multi-step process designed to break down starch in grains to produce simple sugars. Sugars are fermented. Yeast lack enzyme to convert grains to alcohol. Malted barley contains enzyme. Malt and sugars and other adjuvants soaked in warm water, that is termed as mashing. Enzymes in malt act on starch, converting to fermentable starches. Spent grains were removed. Remaining liquid is called wort. Hops are added to wort. Gives beer distinct bitter taste. Also has natural antimicrobial substances. Hops or wort mixture is then boiled. Extract flavors of hops. Convert wort. Concentrate wort, inactivate enzymes and precipitate proteins. Wort is centrifuged to remove solids and cool. Brewer's yeast added to initiate fermentation. Bottom fermenters clump and sink to bottom of fermentation tank. It produces lager beers. Top fermenters distributed throughout and produces other type of beers. Next one is distilled spirits. Fermentation process nearly same as that of beer. Wort is not boiled. Degradation of starch continues through fermentation. When fermentation is complete, alcohol is purified and distilled. Different types of spirits made with different substrates. For example, rum, it is a fermentation of molasses. Whiskey, it is a fermentation of barley. And vinegar, it is an aqueous solution of at least 4% acetic acid, product of oxidation of ethanol. It is strictly aerobic process. Fermentating bacteria or obligate aerobes organisms can tolerate high concentration of acid. Vinegar generator produce available oxygen to hasten oxidation. Sprays alcohol and biofilm of acid bacteria on wood chips. Alcohol trickles and it is oxidized by bacteria. And next one is bread. Bread rises due to carbon dioxide produced through fermentation of sugars by baker's yeast. Any alcohol produced evaporates during baking. Characteristic flavor of sourdough bread is due to addition of lactic acid bacteria to bread making ingredients. Thank you.